Right now, at this very second, behind a secured, heavy door that most patients will never be allowed to open.
Human cells are being kept alive.
Yes, kept alive in an entirely artificial atmosphere. The air inside this room is being scrubbed of microscopic chemical fumes that, well, that you and I cannot even smell.
And the temperature of the surfaces is regulated to within a fraction of a single degree.
Right. And the lighting is deliberately dimmed, simulating the deep internal quiet of the human body.
It is a really profound environment to consider. Because when you remove biological material from the human body, its immediate default trajectory is failure.
Wow, just instant failure.
Exactly. The biological clock begins winding down the absolute second it hits room air. The entire architecture of the space you're describing is engineered to halt that failure.
And that is exactly where we are taking you today. Today's deep dive is a complete, unhurried mapping of the IVF embryology lab.
We're going to step past the clinical waiting rooms.
Right, past the hormone injection schedules and the ultrasound charts that you are probably already familiar with.
We're taking you on a slow, deliberate, and highly mechanical tour of the hidden biology.
From the precise second the raw material is collected to the complex physics of fertilization and freezing, we are serving as your mental orientation to this microscopic world.
And our foundation for this journey relies on a very specific stack of sources.
Yes, we are synthesizing clinical laboratory protocols, advanced embryology workflows, and the biological physics that govern cellular development in vitro.
We are going to intentionally slow our pace today. We want to look through the magnification of the microscopes and really understand the mechanical why and how behind the sequence of events in a complete cycle.
So before we look at a single cell, before we even talk about the biology itself, we have to fundamentally redefine the room.
We do?
Because when I picture a laboratory, I immediately default to the classic chemistry setup. You know, I see the harsh fluorescent lights, the fume hoods, the Bunsen burners.
Right, the chemical regions sitting on cold stainless steel benches.
Exactly. But based on our sources, thinking of the embryology lab in those terms is a complete misconception. This isn't about chemical manipulation at all.
It's a completely different paradigm. The IVF embryology lab is not just a workspace. It is an active, artificial participant in the biological process.
An active participant.
Yes. Its entire structural design is meant to serve as an external extension of the human reproductive tract. The goal is to mimic the physiological conditions of the body with absolute fidelity.
And the margins for error there are microscopically thin, right?
Incredibly thin. Small deviations can halt embryo development entirely. It combines the sterility of an operating theater with the precision of a cell culture facility.
So rather than a traditional chemistry lab, this is more like building a highly secure artificial womb where even the air quality has to be perfect.
That's a great way to picture it.
Let's explore those margins because I want to understand what mimicking the body actually entails from a physics and engineering standpoint. Our sources emphasize temperature first.
Temperature is foundational, yes.
The human body operates at roughly 37 degrees Celsius. But surely a slight fluctuation, say a draft from a door opening, wouldn't be catastrophic to a single cell.
Well, it can be entirely catastrophic.
Yeah.
Particularly for the female reproductive cell, which we must meticulously refer to as the oocyte.
The oocyte.
Yes. The oocyte is perhaps the most temperature-sensitive cell in the entire human body.
Really? Out of all our cells?
Absolutely. Inside that cell, there is a microscopic structural scaffold called the meiotic spindle.
Okay.
It's made of delicate protein fibers called tubulin. And this spindle's job is to hold the cell's chromosomes in perfect alignment, just waiting for the moment of fertilization.
So it's essentially the physical architecture that manages the DNA.
Precisely. And that tubulin structure is highly thermolobable.
Meaning it reacts to heat?
Or the lack of it, yes.
Yeah.
If an oocyte is removed from an incubator and placed on a microscope stage that is even a degree or two cooler, say down to 35 degrees Celsius, that drop in thermal energy causes those spindle fibers to literally depolymerize.
Wait, they dissolve?
They dissolve. The chromosomes lose their anchor, and they scatter chaotically inside the cell.
Oh, wow.
And even if you warm the cell back up to 37 degrees, the spindle may reform abnormally, which leads to a genetically chaotic embryo.
That is astonishing. The literal structural integrity of the DNA relies on ambient thermal kinetic energy.
It does.
Which means the room can't just have a good thermostat on the wall. The microscopes, the glass dishes, the pipettes, Every single physical surface the cell touches must be heated.
Every single surface. They use specialized heated stages on the microscopes, and the ambient temperature of the workstations is tightly controlled.
But temperature is merely the thermal baseline, right?
Yeah.
We also have to consider the chemical atmosphere, the pH of the fluids these cells rest in.
Right, the culture media, the liquid environment.
How does the lab control the acidity or alkalinity of a microscopic droplet of fluid?
They utilize the exact same chemical mechanism your own blood uses, which is a bicarbonate buffering system.
Okay, I think I remember this from biology.
So the culture media contains sodium bicarbonate. But to keep that bicarbonate from shifting the fluid to alkaline, the air inside the incubators is strictly flooded with a specific concentration of carbon dioxide.
A specific concentration.
Usually around 5 to 6%. That CO2 gas actually dissolves into the liquid media droplet, reacting with the water to form a weak carbonic acid.
And that acid balances the bicarbonate.
Exactly.
So the air itself is actively engaged in a chemical reaction with the liquid to hold the pH steady. If the CO2 levels in the incubator fluctuate, the fluid becomes too acidic or too basic.
And if that happens, the cellular enzymes just stop functioning. The cellular metabolism would collapse.
This is why the concept of a test tube baby, or life made in a dish, is a profound misunderstanding of the science.
It's a complete myth. The lab does not manufacture biological life. The lab cannot force a cell to divide.
Right, it's more of an incredibly complex biological sanctuary. The lab is stripping away the hostile variables of the outside world, setting up the exact thermal and chemical stage, and then stepping back to let the inherent genetic potential of the oocyte and the sperm execute their own programming.
It is absolute biological stewardship.
Stewardship, I like that word for it.
And understanding that intense, pressurized stewardship is the only way to fully appreciate what happens the moment the raw biological material actually arrives, the moment the harvest begins.
Let's walk through that arrival.
Yeah.
Because down the hall from this hyper-controlled laboratory is the operating room.
Right.
The clinician is performing the ovum pickup. They are using an ultrasound probe with a long, thin needle attached to it, puncturing the vaginal wall and going directly into the patient's ovaries.
And they apply suction to aspirate the fluid from inside the ovarian follicles.
And this fluid is passed instantly, often through a literal heated hatch in the wall, straight into the lab.
The embryologist is waiting on the other side of that hatch. And it is vital to dispel a common clinical illusion here.
What illusion is that?
Well, the patient might assume that the lab receives a neat, pristine tube containing isolated oocytes. In reality, the embryologist receives test tubes filled with raw, bloody, cloudy follicular fluid.
Wow, okay, so it's the biological equivalent of unrefined ore.
That's a good way to put it. So the embryologist pours this fluid into a heated search dish, places it under the microscope, and they begin scanning.
But they aren't looking for a naked spherical cell, are they?
No, they are scanning the fluid for a structure we call the cumulus oocyte complex.
Cumulus oocyte complex.
Yes, you have to visualize the scale in the architecture here.
Yeah.
The oocyte itself is microscopic, roughly 0.1 millimeters across, but it is entirely encased in a massive, fluffy, gelatinous cloud of thousands of specialized cells.
The cumulus cells. What is their biological purpose? Why is the egg wearing this massive cellular coat?
Well, for months leading up to this moment, those cumulus cells have been the life support system for the oocyte inside the ovary.
Oh, I see.
They are physically connected to the oocyte via microscopic channels, passing nutrients, proteins, and chemical messengers back and forth.
So they're feeding it.
Yes. And they are embedded in a sticky, viscous matrix of hyaluronic acid. When the embryologist looks through the microscope, they aren't seeing the tiny egg. They're looking for the way the microscope's light catches and refracts through this expansive, sticky, cloud-like matrix floating in the blood.
Ah. And once they spot that refractive cloud, the biological clock is ticking loudly, right? Because the fluid is cooling. The pH is shifting because it's no longer inside the body's CO2 environment.
Speed and precision are paramount at this stage.
So what do?
The embryologist uses a specialized glass pipette to physically capture that sticky complex, pulling it out of the bloody fluid. They then transfer it through several separate droplets of clean, warm, buffered culture media.
Like a washing process.
Exactly.
Yeah.
It systematically dilutes and strips away the red blood cells, the inflammatory markers, and the cellular debris until the complex is sitting in pristine fluid. Then it is rushed immediately into the safety of the incubator.
I want to pause the chronological timeline here for just a second and focus on a very difficult mathematical reality that our sources highlight.
Okay.
Because the patient in the operating room went to sleep after seeing their final ultrasound, and that ultrasound might have shown 12 large, beautifully round follicles in the ovaries.
Right.
It is entirely natural for that patient to wake up expecting 12 oocytes.
Yeah.
But the embryologist might walk out of the lab and say, well, we searched every drop of fluid and we only recovered 8 complexes.
It happens often.
Where is the disconnect? Wait, let's unpack this slowly. A patient might see 10 follicles on their ultrasound, but you're saying that doesn't mean the lab will find 10 usable oocytes. The math changes on day one.
The math absolutely changes on day one, and the disconnect lies in the limitation of ultrasound technology versus microscopic reality.
How?
An ultrasound utilizes sound waves to create a shadow picture of fluid density. When a clinician measures a follicle on a screen, they are merely measuring a sac of fluid. They cannot see the microscopic oocyte anchored to the inner wall of that sac.
So a beautifully large follicle could simply be a biological empty shell.
It happens frequently.
Yeah.
Or, equally as common, the biological mechanism required to release the cell fails.
The release mechanism.
Yes. 36 hours prior to the surgery, the patient takes a trigger medication, usually a hormone called HCG. This hormone's job is to instruct the cumulus oocyte complex to physically detach from the follicle wall so it floats freely in the fluid.
So it can be vacuumed up.
Exactly. But if the receptor density in a specific follicle is poor or the chemical signal doesn't fully penetrate, the complex remains stubbornly glued to the ovarian wall.
Oh, I see.
When the clinician's needle vacuums out the fluid leaves, but the complex stays behind.
That is a harsh biological reality. The aspiration procedure collects the fluid, which represents the opportunity for a cell, but it does not guarantee the certainty of one. The numbers funnel begins to narrow violently from the very first hour.
The lab can only protect what biology successfully yields. And while the embryologist is washing and securing that female genetic yield in the incubators, an entirely parallel, equally complex process is happening simultaneously.
The other half of the equation, the processing of the male genetic contribution, the sperm.
Yes.
Our sources dictate this happens on the exact same timeline. The semen sample is collected and arrives at the lab. And my initial thought, based purely on natural conception, is that you would simply introduce a few drops of that raw sample into the dish with the eggs.
That would be the intuitive thought, yes.
Right. Nature does it raw. Why wouldn't the lab?
Well, attempting that in the lab would be lethal to the oocytes. We must remember that natural, in vivo reproduction involves a massive, hostile obstacle course. The human vagina, the cervix, the uterus, the fallopian tubes, they are not just pathways. They are intense biological filters.
Filters designed to strip away everything except the genetic payload.
Correct. Raw semen is predominantly composed of seminal plasma, which is a complex cocktail of prostaglandins, zinc, proteolytic enzymes, and fructose.
Okay.
In the body, this plasma is left behind in the vagina, while the sperm swim up through the cervical mucus.
So the plasma never reaches the egg.
Exactly. If you were to drop raw seminal plasma directly onto an oocyte in a sealed culture dish, those enzymes and prostaglandins would quickly degrade the culture media, induce overwhelming oxidative stress, and kill the oocyte.
Wow.
Furthermore, raw semen contains dead sperm, white blood cells, and bacteria.
So the embryologist has to artificially replicate the filtration of the human cervix and uterus. They have to extract only the motile, structurally sound sperm and leave the toxic plasma behind.
They do.
But how do you physically separate microscopic swimming cells from the liquid they are suspended in without crushing them?
The lab employs the laws of physics, specifically fluid dynamics and centrifugal force. The primary method, detailed in our clinical protocols, is called density gradient centrifugation.
Let's break down the mechanics of that. A centrifuge spins samples at high speeds. But what is the density gradient aspect?
The embryologist takes a conical test tube and carefully layers specialized liquids inside it. These liquids are made of colloidal silica particles, coated in saline.
Okay, so it's a very specific chemical liquid.
Yes, they are biologically inert, but they are incredibly dense. The embryologist places a thick, heavy layer at the bottom of the tube, say a 90% density solution.
Right.
On top of that, they gently float a slightly thinner layer, a 45% density solution. Finally, they pipette the raw liquefied semen sample to rest on the very top.
It's like a microscopic hydrostatic obstacle course. You have the raw sample at the top, a thick liquid barrier in the middle, and an incredibly dense liquid barrier at the bottom.
Exactly the right framing. When this tube is placed into the centrifuge and spun, massive gravitational forces are applied. The cells are pulled downward.
But they encounter the dense silica liquids.
Yes. And the seminal plasma, the bacteria, and the cellular debris do not have the molecular density to push through the barrier. They remain trapped at the top.
And the sperm, what happens to them?
This is where morphological physics comes into play. A sperm cell with a highly condensed, perfectly packaged DNA head and a strong, structurally sound tail has a very high specific gravity.
It's heavy.
It acts like a microscopic torpedo. As the centrifuge spins, only the most structurally perfect dense sperm have the kinetic mass to pierce through the 45% layer and then push all the way through the incredibly thick 90% layer, collecting at the very bottom of the tube in a tiny purified pellet.
And the malformed ones?
Sperm that are malformed or have vacuoles in their heads lack the density and get caught in the upper layers.
That is brilliant. You are using the physical gravity of healthy DNA packaging to filter out the structurally weak cells.
Yes, it is highly elegant.
And our sources mention an alternative method, too, the swim-up technique. That sounds less about gravity and more about pure kinetic energy.
The swim-up relies entirely on the sperm's inherent motility, its rheotaxis, or forward swimming ability. The embryologist takes the semen sample and gently layers highly nutritious, clean culture media directly on top of it.
Do they spin it?
They do not spin it. They simply place the tube in the incubator at a 45-degree angle.
And wait for the biology to declare itself?
The dead sperm and the debris will sit passively in the bottom layer. But the highly motile, energetic sperm will actively swim upward out of the seminal plasma and into the clean nutrient media.
Ah, so they literally swim up out of the muck.
After an hour, the embryologist simply skims the top layer of fluid. What they harvest is an elite, self-selected population of the most kinetically active sperm.
So by noon on day 0, the lab has successfully curated both halves of the equation. The oocytes are washed and resting in their pH-balanced, temperature-locked incubators. And the sperm have been physically and kinetically filtered, stripped of all toxic plasma. This brings the embryology team to the first major strategic crossroads of the cycle, the fertilization decision.
The clinical protocols dictate two divergent pathways for fertilization: conventional in vitro fertilization, or ICSI.
Let's examine conventional IVF first. Because despite the hyper-advanced setting, this pathway leans heavily on the ancient organic mechanics of biology. What does conventional mingling actually entail on a microscopic level?
The embryologist creates a small droplet of culture media in a dish. They place the cumulus oocyte complex into that droplet.
And remember, the oocyte is still wearing that massive, sticky cloud of cumulus cells at this point.
Yes, exactly. Then, the embryologist calculates a very specific concentration of the purified sperm, usually tens of thousands of motile sperm, and introduces them into the same droplet.
And then they just close the incubator door.
You step away entirely. The biological mechanisms must take over. The sperm must sense the chemical attractants released by the oocyte. They must swim vigorously toward the complex.
And when they reach it?
When they hit that sticky cumulus cloud, they release an enzyme from the tip of their heads called hyaluronidase. This enzyme chemically dissolves the hyaluronic acid, binding the cumulus cells together.
So thousands of sperm are collectively tunneling through the cloud, using their enzymes to pave a road to the center.
It is a massive collective effort. But once a sperm reaches the cloud, it encounters the ultimate physical barrier, the zona pellucida.
What is that?
This is a thick, protein-based shell directly encasing the oocyte. The sperm must physically bind to specific glycoprotein receptors on this shell, known as ZP3 receptors.
Like a lock and key.
Very much so. Binding triggers the acrosome reaction, where the sperm violently dumps the rest of its enzymes to burn a microscopic hole through the shell, thrusting its head inside to fuse with the oocyte membrane.
Conventional IVF requires every single step of that natural biochemical cascade to fire perfectly.
Every single step.
The motility must be there, the enzyme release must be timed perfectly, and the receptor binding must match.
It demands profound biological competence from both the sperm and the egg. But the data reveals a critical vulnerability here.
Right. What if the male patient has severe oligospermia, a critically low sperm count?
Or what if the microscopic anatomy of the sperm reveals severe morphological defects, meaning they physically cannot tunnel through the zona pellucida?
If they lack the kinetic energy or the enzymatic keys, conventional IVF would result in a total fertilization failure. You'd check the dish the next day, and the eggs would still be sitting there untouched.
Yes, which necessitates the second pathway, the micromanipulation technique known as ICSI, intracytoplasmic sperm injection.
ICSI. This is perhaps the most dramatic intervention in the embryology workflow. In this pathway, we completely abandon the concept of the sperm naturally tunneling through the barriers.
Yes, we do. But to inject a sperm directly into an oocyte, the embryologist must first clear the visual field. They cannot inject through the massive, sticky cumulus cloud.
Right, they have to physically dismantle the life support system. How do you strip away thousands of sticky cells without crushing the microscopic egg inside?
We use the exact same enzyme the sperm uses, but we deliver it artificially.
Oh, that's clever.
The embryologist bathes the complex in a concentrated solution of synthetic hyaluronidase. This biochemically dissolves the bonds between the cumulus cells. Then, using a microscopic glass pipette with a diameter barely larger than the oocyte itself, they gently draw the cell in and out.
Just moving it back and forth.
Yes. The sheer force of the fluid moving through the narrow glass cube mechanically strips away the remaining loosened cumulus cells.
And stripping away the cloud reveals the naked oocyte. But stripping it naked serves a dual purpose, right? It's not just to clear the path for the injection needle. It's because the embryologist must perform a critical visual inspection. They have to assess the maturity of the cell.
This is a fundamental concept in reproductive biology. When an oocyte is inside the body, it is genetically paused in an immature state.
Okay, meaning what?
It holds a full set of 46 chromosomes. But to combine with the sperm, which brings 23 chromosomes, the oocyte must discard half of its own DNA. It must undergo meiosis and drop down to 23 chromosomes.
If it doesn't drop half its DNA, combining with the sperm would create a cell with far too many chromosomes, which is biologically fatal.
Absolutely fatal.
So how does the embryologist looking through a microscope know if this invisible genetic reduction has actually happened?
They look for the physical garbage bag of discarded DNA.
The garbage bag.
Yes. When the oocyte correctly halves its chromosomes, it packages the discarded 23 chromosomes into a tiny, distinct bubble of membrane and pushes it outside of its own cellular cytoplasm, pinning it just beneath the outer zone of pellucida shell. We call this tiny bubble the first polar body.
Ah. So if the embryologist looks at the naked oocyte and sees that tiny extra bubble resting on the surface, they know the cell has successfully discarded its extra DNA.
That specific structural stage, possessing a single polar body, is clinically defined as M2, metaphase 2. Only an M2 oocyte is biologically competent to accept a sperm. If the embryologist looks and there is no polar body, the cell is stuck in the immature M1 stage, metaphase 1.
And an M1 cell still has all 46 chromosomes.
Right. You cannot perform ICSI on an M1 oocyte. The needle would likely rupture the fragile, unorganized genetic spindle inside.
Let's follow the M2 cell. It has the polar body. It is mature. It is ready. The embryologist moves this naked oocyte to a micromanipulator.
Yes, this is an advanced microscope outfitted with pneumatic or hydraulic joysticks that translate large hand movements into microscopic, nanometer-level movements of glass tools.
The choreography of ICSI is mesmerizing. The embryologist navigates over to a droplet containing the purified sperm. Under 400x magnification, they scan the swimming cells.
They are performing visual genetic selection here. They're looking for one single sperm with a perfectly oval head, no vacuoles, a straight mid-piece, and a vigorous swimming pattern.
Once they identify the target, they have to catch it. But you can't inject a wildly thrashing sperm into an egg, right? It would tear the egg's internal architecture apart.
The motility must be neutralized. The embryologist uses an incredibly sharp, hollow glass injection needle. They bring the needle down directly over the sperm's tail and rapidly strike it against the bottom of the plastic dish.
Oh, wow. They hit it.
Yes, this microscopic trauma breaks the membrane of the tail, permanently immobilizing the sperm. It also triggers an influx of calcium into the sperm, which actually helps activate it later.
So they aspirate that single paralyzed sperm into the hollow tip of the glass needle. Then they use the joysticks to navigate back over to the droplet holding the M2 oocyte.
Using a blunt holding pipette, applying gentle suction, they grasp the left side of the oocyte to lock it in place. Crucially, they rotate the oocyte, so the polar body is resting at the 12 o'clock or 6 o'clock position. The injection needle will enter from 3 o'clock.
Why does the positioning of the polar body matter?
Well, the polar body indicates the location of the meiotic spindle. Remember that fragile web of tubulin holding the maternal DNA?
Right, from earlier.
By keeping the polar body at 12 or 6, the embryologist ensures the needle, entering at 3 o'clock, drives into the empty cytoplasm, avoiding and protecting the maternal DNA from being physically crushed by the glass.
That level of geographic precision is incredible. The needle drives through the thick outer shell, the zona pellucida. But breaking the shell doesn't mean you are inside the cell yet. There is an elastic membrane beneath it, the ulema.
The ulema is highly elastic. The needle pushes into it like a finger pushing into a balloon without popping it.
How do they get through?
To successfully breach the membrane, the embryologist must apply gentle negative pressure. They pull a tiny bit of the egg's internal cytoplasm back into the needle until the membrane finally snaps.
That sudden snap indicates open access to the interior.
Yes. They then smoothly reverse the pressure, depositing the single sperm directly into the center of the cell, and slowly withdraw the needle.
To visualize this, conventional IVF is like putting everyone in a room and letting biology take its course organically. ICSI is like a highly orchestrated blind date, where the lab plays matchmaker with a single specific sperm.
Exactly.
It's an incredibly invasive mechanical bypass of natural selection. And while watching ICSI on a monitor looks like a guaranteed success, you literally put the DNA inside the cell. Our sources emphasize a critical caveat. Delivering the payload does not equate to a successful biological detonation.
That is the most persistent myth in the IVF lexicon. ICSI guarantees penetration. It does absolutely nothing to guarantee fertilization.
Wait, really? It doesn't guarantee it.
Not at all. Once the embryologist withdraws the needle and returns the dish to the incubator, the mechanical phase ends. The biochemical cascade must begin.
The natural cascade.
Yes. The sperm head, which is tightly compacted with specialized proteins called protamines, must entirely unravel and decondense. The oocyte must release waves of calcium to activate and resume its genetic cycle. The maternal and paternal DNA must physically find each other and organize a new mitotic spindle.
The lab can force the two puzzle pieces into the same box, but it cannot force the pieces to physically lock together. If the chemical signaling is deficient, the biology stalls.
The cell will simply arrest.
Which brings us to the tension of the following morning. Day 1. 16 to 18 hours after the sperm and the egg were introduced, whether by the organic mingling of conventional IVF or the sharp precision of ICSI, the embryologist must assess the outcome.
They must determine if the genetic cascade actually fired. This checkpoint is known as the fertilization check, and it is highly time-sensitive.
Why is it so time-sensitive?
If you look too early, the biological structures haven't formed yet. If you look too late, the structures will have vanished as the cell prepares for its first division.
So the embryologist pulls the dish from the incubator, places it under the microscope, and looks at the single cell. What specific structural evidence indicates that fertilization was successful?
They are looking deep within the cytoplasm for the appearance of two distinct spherical structures called pronuclei.
Pronuclei.
Yes. When the maternal DNA finishes its meiosis, it wraps itself in a temporary nuclear envelope, the maternal pronucleus. Simultaneously, the decondensed sperm DNA wraps itself in its own envelope, the paternal pronucleus.
And what do these spheres do?
These two spheres migrate to the center of the cell and rest directly beside each other, their membranes nearly touching.
Like two blueprints sitting on a table waiting to be combined.
Exactly the right visualization. Seeing those two spheres, a status clinically recorded as 2PN or 2 pronuclei, is the definitive hallmark of normal, successful fertilization. It confirms that the male and female genetics are present and organizing correctly.
But the clinical notes dictate that the embryologist is also screening for failure. What structural anomalies reveal that the biological cascade went wrong?
Frequently, the embryologist will look at a cell and see 0PN, no pronuclei.
Meaning nothing happened.
The cell is visually identical to how it looked the day before. This means fertilization completely failed. The calcium waves didn't fire, the sperm didn't decondense, the engine simply flooded and never turned over.
And what if they see just one? A 1PN cell?
1PN is a complex anomaly. It can mean parthenogenesis occurred, where the oocyte activated itself due to the mechanical trauma of the needle or environmental stress, forming only a maternal pronucleus without the sperm's contribution.
Ooh.
Or it could mean the sperm is inside, but it completely failed to decondense and form its own envelope. Regardless of the mechanism, a 1PN cell is genetically imbalanced and generally considered non-viable.
The sources also mention the phenomenon of polyspermy, which presents visually as 3PN, 3 pronuclei. How does a cell end up with 3 sets of DNA?
In conventional IVF, polyspermy occurs when the biological defense mechanisms fail. Normally, the exact microsecond a single sperm breaches the membrane, the oocyte triggers a cortical reaction.
What does that do?
It dumps thousands of tiny granules into the space beneath the shell, instantly hardening the zona pellucida into an impenetrable concrete wall, blocking any other sperm from entering.
A microscopic lockdown.
But if that reaction is delayed by even a fraction of a second, 2 separate sperm can breach the membrane simultaneously. Both decondense. Both form pronuclei. The result is a 3PN cell holding 69 chromosomes instead of the requisite 46.
Wow.
It is fundamentally chaotic and completely incompatible with life.
So this day one check is a brutal funnel. The 0 PNs, 1 PNs, and 3 PNs are discarded. Only the perfect 2 PN cells are kept. But I want to linger on the psychological weight of the 2 PN cells. For a patient, hearing that fertilization was successful feels like crossing the finish line, but the biochemical reality is far more sobering.
The clinical reality is that 2PN only confirms the blueprints have been drafted and brought to the construction site. It offers zero guarantees that the cellular machinery is capable of actually building the house.
Right.
The maternal and paternal DNA still have to physically merge, a process called syngamy, and initiate the first cellular division.
And that transition from a single cell holding the blueprints to a multicellular organism actively building itself happens entirely in the dark. This marks the beginning of the culture phase, the hidden days.
The most vulnerable phase of the entire process. After the day one check, the newly confirmed embryos are returned to the incubators. They will remain there for 5 to 7 days, and the environment they are cultured in is a marvel of biological physics.
Let's examine the physics of the culture dish. Because the embryos aren't just swimming in a giant beaker of fluid, they are housed in micro droplets of highly specialized culture media. And the protocols note that these droplets are submerged under a layer of oil. Mineral oil.
Yes, mineral oil.
Why introduce an industrial-sounding substance like oil to a delicate biological system?
It is an elegant solution to a fatal physical problem, which is evaporation. The incubators are heated to 37 degrees. If you place a 25 microliter droplet of water-based media on a plastic dish in that heat, the water will evaporate almost instantly.
And if the water evaporates, the salts and proteins dissolved in that media are left behind.
They become hyperconcentrated. The osmolality of the droplet spikes exponentially. This creates a massive osmotic gradient, which literally sucks the water out from inside the embryo cells, collapsing them and killing the embryo.
So how does the oil prevent that?
Well, mineral oil is hydrophobic and completely immiscible with the water-based media. By gently flooding the dish with a layer of oil, the embryologist creates an impenetrable physical seal over the media droplets.
A liquid seal.
The oil locks the water molecules in, preventing evaporation and maintaining perfect osmolality. It also acts as a thermal buffer against sudden temperature drops when the dish is removed from the incubator, and it shields the media from airborne contaminants.
It creates a perfectly isolated, stable microuniverse for every individual embryo. And the atmosphere inside the incubator itself is heavily modified. The air we breathe in a room is roughly 20% oxygen, but the lab purposefully suppresses the oxygen inside the incubators down to around 5%.
Yes.
If oxygen is the fuel of life, why starve the developing embryo of it?
We have to look back at the physiological reality of the human body. Deep inside the fallopian tubes and the uterus, the oxygen tension is incredibly low. It is a naturally hypoxic environment.
Oh, I hadn't thought of that.
If you expose a fragile, dividing embryo to 20% atmospheric oxygen, the cellular metabolism generates excessive reactive oxygen species.
Free radicals?
Yes. Free radicals are highly unstable molecules that aggressively strip electrons from surrounding structures. They cause massive oxidative stress, literally shredding the embryo's DNA, damaging the mitochondrial membranes, and triggering cellular suicide or apoptosis.
Wow, oxygen is actually toxic to them at that level.
It is. By lowering the oxygen to 5%, the lab mimics the dark hypoxic safety of the reproductive tract, forcing the embryo to utilize anaerobic metabolic pathways that generate far less oxidative damage.
So they are resting in the dark under oil in a hypoxic atmosphere, and the embryologist must periodically pull them out to assess their progress. The protocols highlight 2 major assessment checkpoints, day 3 and day 5.
Correct.
Let's look at day 3. This is the cleavage stage. The single cell should have divided into multiple cells. What specifically is the embryologist measuring when they look through the microscope?
On day 3, the embryo should ideally consist of roughly 6 to 10 cells called blastomeres. The embryologist is assessing the mechanical efficiency of cell division. First, they evaluate symmetry. Are all the cells perfectly equal in size?
Why does size symmetry matter?
Because cell division should be an equal partitioning of cytoplasm. If an embryo divides unevenly, creating one massive cell and several tiny ones, it indicates that the internal structural proteins driving the division are flawed or chaotic.
The machinery is stuttering.
Exactly. The second primary metric on day 3 is fragmentation. As cells divide, sometimes the outer membrane pinches off tiny, nucleus-free bubbles of cytoplasm. These are fragments. Think of them as cellular debris or biological sawdust left over from a messy cut.
Is any amount of fragmentation acceptable, or is it a sign of impending failure?
A very low degree of fragmentation, say under 10% of the total volume, is common and often benign. But heavy fragmentation is highly detrimental.
Because it takes up space.
Yes. Those useless bubbles of debris physically take up space inside the outer shell. They physically block the healthy cells from touching and communicating with one another. If the cells cannot form tight junctions and signal each other, the embryo will eventually arrest.
So day 3 is an assessment of the basic structural plumbing. But day 5, this represents a massive, complex biological shift. This is the blastocyst stage. The embryo isn't just a cluster of identical cells anymore.
Day 5 is the defining morphological milestone of pre-implantation development. Around day 4, the embryo undergoes a process called compaction. The outer cells form incredibly tight junctions, sealing the borders.
Sealing themselves up.
Then they begin pumping fluid into the center of the embryo. This hydraulic pressure creates a large fluid-filled cavity called the blastocyst.
The physical pressure forces the cells to organize and differentiate.
They differentiate into two entirely distinct, highly specialized lineages. The first is a tight, dense cluster of cells that is pushed to one side of the cavity. This is the inner cell mass.
And the clinical destiny of the inner cell mass.
Those specific cells will continue to divide and eventually fold into the embryonic disc, ultimately becoming the fetus itself.
Wow.
The embryologist grades this cluster heavily. They want to see a large, tightly packed mass containing dozens of robust cells. A sparse, loose inner cell mass is a poor prognostic indicator.
And the second lineage of cells?
The cells that were pushed to the perimeter, forming the outer sphere holding the fluid. That single layer of cells is the trophectoderm.
The trophectoderm is what will eventually attach to the uterine wall and become the placenta.
It is the biological life support and invasion system. The embryologist grades the trophectoderm based on cell count and cohesion. You want a continuous, strong layer of many cells.
And if it's sparse?
If there are very few cells stretched thin over the cavity, the embryo will lack the biological machinery to successfully implant into the endometrium.
The grading system evaluates the size of the cavity, the density of the fetal cells, and the integrity of the placental cells. It sounds incredibly comprehensive.
It is very detailed.
But I want to introduce a major pushback here, based on the expert notes in our outline. If grading is based on how they look, how do you know a pretty embryo is actually healthy? Is morphology ever deceiving?
It absolutely can be, and it does so frequently. This is perhaps the most profound limitation in modern embryology. Morphology is a highly useful selection tool, but it is not a guarantee of genetic destiny.
Break down the biology of that deception. How can an embryo look perfect but fail?
An embryo can execute the mechanics of cell division flawlessly. The tubulin spindles pull the cells apart, the tight junctions form perfectly, the fluid cavity expands beautifully. Under the microscope, it receives a perfect AA grade. It looks stunning.
But inside?
Yet, inside the nucleus of those beautiful cells, the chromosomes could be entirely chaotic.
Yeah.
Instead of 46 chromosomes, it might have 45 or 47. This is called aneuploidy.
So the structural proteins building the house are working fine, but the architectural blueprints inside the walls are catastrophically wrong.
The microscope cannot see the DNA. A visually perfect embryo with an aneuploid genetic payload will almost certainly fail to implant or result in an early miscarriage.
And the reverse must be true as well, right?
Conversely, an embryo might struggle mechanically. It might divide a little slowly. Its inner cell mass might look slightly disorganized or modest in size. It receives a BC grade. But its genetics are absolutely perfect. That modest-looking embryo has the capacity to result in a completely healthy live birth.
The visual assessment is the best non-invasive tool available, but it is fundamentally blind to the chromosomal reality. Which brings us to the culmination of the laboratory phase. The embryos that have survived the gauntlet of culture and grading must now exit the system.
Yes, they have to move on.
Our sources outline 3 distinct downstream pathways: the fresh transfer, cryopreservation, and pre-implantation genetic testing. Let's start with the immediate clinical route, the fresh transfer.
A fresh transfer occurs just 3 to 5 days after the initial egg retrieval. It requires the patient's uterine environment to be clinically synchronized with the embryo's development.
So timing is everything.
If the hormone levels are optimal and the endometrial lining is receptive, the embryologist prepares the selected embryo for transfer.
The logistics of this must be incredibly fraught. You are moving a microscopic, invisible entity from a secure dish into a dark biological cavity. How do they ensure the embryo doesn't simply get lost or stuck in the equipment?
It relies on intense fluid dynamics and rigorous witnessing protocols. The embryologist uses an incredibly thin, flexible catheter. Working under the microscope, they aspirate a tiny amount of culture media, then a small air bubble, then the microscopic droplet containing the embryo, then another air bubble, and finally a cap of media.
The air bubbles act as physical bumpers, keeping the embryo isolated and visually marking its position in the fluid column.
Precisely. Before this catheter ever leaves the lab, an ironclad chain of custody is executed. The identity of the dish, the catheter, the patient waiting in the transfer room are verbally matched and frequently verified via electronic RFID tags.
There is absolute zero tolerance for error.
None whatsoever.
The clinician guides the catheter into the uterus and gently depresses the syringe, expelling the fluid column. But the procedure isn't over when the syringe is empty, is it?
The most critical step happens post-transfer. The clinician carefully withdraws the empty catheter and hands it immediately back to the embryologist. The embryologist rushes it back to the microscope and flushes clean media through the entire length of the tube, meticulously scanning the fluid.
They have to physically verify that the embryo didn't get stuck to the plastic wall of the catheter.
They must visually confirm the absence of the embryo. Only when the embryologist calls out that the catheter is clear is the transfer officially considered complete.
The physics of the fresh transfer are tense, but the physics of the second pathway, cryopreservation, are mind-bending.
It is remarkable science.
Because if the patient's body is too stressed from the hormone medications, or if there are surplus high-quality embryos, the lab must freeze them. But water is the enemy of freezing. If I freeze a strawberry, the water inside its cells expands, forms jagged ice crystals, and shreds the cellular membranes. When it thaws, it turns to mush.
Right.
How do you freeze a human embryo without the water crystals instantly destroying it?
You utilize a technique called vitrification. It is essentially flash freezing, but it relies on complex biochemistry to avoid ice formation. The embryologist cannot simply plunge the embryo into liquid nitrogen. First, they must dehydrate it.
They have to remove the water. But if you suck all the water out of a cell, the internal salts become so concentrated that they denature the proteins.
That is the biological paradox of freezing.
Mm.
The solution is to replace the water with a highly concentrated cryoprotectant, molecules like dimethyl sulfoxide or ethylene glycol. These act as biological antifreeze.
Oh, antifreeze.
The embryologist moves the embryo through a series of droplets with increasingly high concentrations of these cryoprotectants. The chemicals permeate the cell membrane, pulling the water out and replacing it.
They are essentially filling the cell's plumbing with antifreeze.
Once the cell is permeated, the speed of cooling is the final critical factor. The embryo is loaded onto the microscopic tip of a plastic wand in a volume of liquid so small it is nearly invisible to the naked eye. The embryologist plunges that wand directly into liquid nitrogen at minus 196 degrees Celsius.
Minus 196 degrees. The temperature drop must be instantaneous.
The cooling rate is roughly 20,000 degrees per minute. Because the cryoprotectants have made the internal fluid so incredibly viscous, and because the temperature drop is so violently fast, the molecules literally do not have the time to organize themselves into a crystalline lattice.
So no ice forms.
Exactly. They freeze in place instantly, forming a solid, amorphous liquid, a glass-like state.
Vitrification literally translates to turning to glass. The biological clock doesn't just slow down. Kinetic energy drops so low that time entirely stops.
The embryo is suspended in absolute biological stasis, and our sources point out a major shift in clinical strategy here.
What's the shift?
Historically, freezing was a backup plan for leftover embryos. Today, many clinics utilize an intentional freeze-all strategy. The high doses of hormones used to stimulate the ovaries can make the uterine lining incredibly hostile to implantation.
Right, the body is overwhelmed.
By vitrifying all the embryos, the clinical team allows the patient's body to completely flush out the medications, returning to a calm, natural physiological baseline.
A month later, you thaw the embryo into a perfectly receptive, natural environment. Freezing isn't just a storage method. It is a primary clinical tool to optimize the timing of implantation.
Yes.
Which directly links to the third most advanced pathway, pre-implantation genetic testing, or PGT.
PGT resolves the morphological deception we discussed earlier. It allows us to look past the physical beauty of the blastocyst and count the actual chromosomes. But to test the DNA, we must extract cells from the living embryo.
How does an embryologist physically amputate cells from a microscopic, fluid-filled sphere without collapsing the entire structure or killing it?
It requires extreme micromanipulation and the use of an infrared diode laser. On day 5 or day 6, when the blastocyst is fully expanded, the embryologist uses a holding pipette to secure the embryo.
Where do they cut?
They ensure the inner cell mass, the fetal cells, is positioned safely on the far side of the embryo, away from the working area.
They only target the trophectoderm, the future placenta.
Correct. They use a glass biopsy pipette to apply gentle suction to a small cluster of 3 to 5 trophectoderm cells, pulling them slightly away from the main sphere. Then, using the micromanipulator, they fire incredibly short, microsecond pulses of the laser at the junctions connecting those cells.
When I think of a laser, I think of thermal heat. How does the laser not instantly cook the cells it touches?
The laser is tuned to a very specific wavelength that targets the protein bonds of the cellular junctions, not the water inside the cells. The pulses are so infinitesimally short that the thermal relaxation time of the tissue prevents the heat from spreading.
So it's more like a scalpel.
Exactly. The laser acts as a microscopic scalpel, ablating the bonds until the small cluster of cells breaks free.
The biopsied cells are placed in a tube and sent to a specialized genetics lab where the DNA is amplified and sequenced to count the chromosomes. But sequencing takes days, sometimes weeks. The embryo can't just sit on the lab bench waiting for the results.
This is why PGT relies entirely on the perfection of vitrification. The moment the biopsy is complete, the biopsied embryo is flash frozen into that glass-like stasis.
And it waits.
It waits safely and the liquid nitrogen endures. When the genetic report returns, the clinical team can match the frozen embryo's ID to the results, ensuring that only a chromosomally normal, perfectly competent embryo is eventually thawed and transferred.
The sequence of events we have just mapped, from the frantic, temperature-critical search for the cumulus cloud, to the hydrostatic filtering of the sperm, to the mechanical piercing of the ICSI needle, the physics of the oil-covered culture droplets, and the laser ablation of placental cells, it is an overwhelmingly complex chain of mechanical and biological interventions.
It is truly staggering when you look at it end to end.
But our sources dedicate a significant portion of their protocols to an aspect of the lab that has nothing to do with microscopes of biology.
They focus heavily on the unseen backbone of the lab, the quality control systems, because none of the biological marvels you discussed matter if the foundational environment fails.
So the excellence of an IVF lab isn't just in the steady hands of the embryologist, it's in the invisible systems designed to prevent the wrong thing from happening.
Precisely. We like to romanticize the embryologist as a lone artisan with incredibly steady hands, but the reality is that clinical excellence is driven by ruthless, invisible systems designed to catch human error and mechanical drift before they cause harm.
You mentioned electronic witnessing earlier. How pervasive is that in the workflow?
It is ubiquitous. Every dish, every tube, and every cryo wand is embedded with a microchip or a sophisticated barcode. The workstations are equipped with sensors.
How does that work in practice?
If an embryologist attempts to bring a dish containing patient A's sperm into the same workspace as a dish containing patient B's oocytes, the system instantly triggers an auditory and visual alarm, physically halting the work.
It's an electronic interlocking system enforcing the chain of custody. And what about the environmental stability? Earlier, we talked about carbon dioxide levels regulating the pH. How does the lab guarantee those gas levels never drift?
Every incubator is hardwired into continuous monitoring software. If the CO2 level fluctuates by even 0.1%, or if the temperature of a heated stage drops by 0.5 degree, the system sends immediate alerts to the embryologist's phones 24 hours a day.
So they're constantly monitored.
The liquid nitrogen storage tanks holding the frozen embryos are outfitted with weight sensors and thermal probes. If the nitrogen begins to evaporate faster than expected, alarms cascade through the facility.
Our sources also highlight a massive infrastructural focus on volatile organic compounds, VOCs.
This is perhaps the most critical yet invisible threat to in vitro culture. VOCs are microscopic airborne chemicals.
Like what?
The fumes from floor wax, the off-gassing of the plastic used to make the petri dishes, the propellant in a clinician's deodorant, or even the exhaust from traffic outside the building. To an adult human, these trace compounds are undetectable and harmless.
But to an embryo?
To an embryo developing outside the protective barriers of the human body, VOCs are lethal. They dissolve instantly into the culture media droplets, alter the pH, and cause profound metabolic toxicity, halting cell division.
How do you build a room that keeps out fumes from the street outside?
The entire HVAC system of an IVF lab is bespoke. The air is pushed through massive banks of activated carbon and HEPA filters to physically scrub the VOCs. Furthermore, the lab is maintained under positive pressure.
Positive pressure meaning there is more air pumped into the lab than into the surrounding hallways.
Exactly. When the heavy laboratory doors are opened to receive a fluid sample from the operating room, the clean, pressurized air forcefully pushes outward into the hallway. The dirty, unscrubbed hallway air physically cannot enter the lab. The environment defends itself.
The level of intense paranoid engineering required to sustain this microscopic biology is staggering. It brings us back to the intersection where this entire journey takes place.
The lab sits at the absolute intersection of profound human hope and cold, unforgiving biological precision. Biology, timing, and clinical context all merge here. The laboratory provides the ultimate, optimized stage. It perfects the lighting, stabilizes the temperature, and strips away the toxic variables.
But it cannot force the actors to perform.
No, it cannot rewrite a chaotic genetic script. The embryologist watches, protects, intervenes when mechanically necessary, and ultimately guides the biology as far as its inherent limits allow.
That distinction between stewardship and manufacturing is the most vital takeaway of this entire map.
And as we conclude this foundational orientation to the lab environment, I want to leave you, the listener, with a forward-looking paradigm shift. A concept to ponder that builds on everything we've discussed about assessing these fragile embryos. If the lab's goal is to perfectly mimic the human body's conditions, what if the future of embryology isn't about better microscopes?
Let's explore it.
Currently, our entire assessment framework relies heavily on visual disruption. We look at the physical architecture of the cells under the microscope, or we fire a laser to physically sever pieces of the trophectoderm for genetic testing. But what if the next massive evolution doesn't rely on looking or cutting at all? What if it relies on deciphering the invisible chemical language the embryo is already speaking?
What do you mean by chemical language?
Well, remember the tiny microdroplet of culture media. The embryo spends 5 days growing inside. As that embryo divides, it consumes glucose, pyruvate, and amino acids from the fluid. And crucially, it secretes its own metabolic waste products and microRNA back into that exact same droplet.
It's leaving a microscopic chemical footprint in the water.
A highly specific, complex metabolic signature. The emerging field of non-invasive metabolomics asks a profound question. What if we could extract that tiny droplet of leftover fluid after the embryo has been frozen and run it through a mass spectrometer?
To see what's in it.
Yes. What if we could read the exact ratio of consumed nutrients and secreted proteins and use that chemical signature to determine exactly how viable the embryo is or exactly what chromosomes it holds without ever needing to touch the embryo with a laser?
So instead of physically biopsying the house to see if the wiring is sound, you just analyze the exhaust coming out of the chimney.
It would completely eliminate the mechanical stress of the biopsy. It would shift the lab from a paradigm of invasive testing to a paradigm of silent chemical observation. We are moving toward a future where we simply learn to listen to what the cells are already telling us.
Deciphering the microscopic exhaust of a dividing cell to predict its viability, that is an absolutely brilliant concept to mull over.
It changes the entire biological approach.
It completely reframes the future of the laboratory. Thank you for joining us today on this incredibly deep, deliberate journey. By building this comprehensive mental map of the IVF embryology lab, by understanding the physics of the incubators, the intense biomechanics of fertilization, and the incredible engineering behind cryopreservation, it entirely demystifies the black box of the process.
It transforms the invisible sequence into a highly logical, structured reality.
It absolutely does. And now that we have constructed this foundational map together, our future deep dives are going to zoom in even further. We are going to unpack each specific procedure we touched on today in exacting, granular detail. And we will be starting next time at the very beginning, exploring the complex fluid dynamics and the intense microscopic search of the ovum pickup.
We look forward to having you with us for that exploration.
Until next time, remember that the laboratory does not manufacture life. It fiercely protects the sanctuary where inherent biology is allowed to unfold. Keep exploring.